Today, Professor Dial will talk about assessment of speech perception and language comprehension in aphasia using temporal response function modeling. And the floor is all yours, Heather.
Okay, thank you. So there we go. Kind of notorious for unreasonably long titles. And here is my unreasonably long title. I'll jump right in. So I'm going to talk today about speech perception and language comprehension in aphasia and how we are using computational modeling to assess these different processes. So I'm going to start by talking about processing of the speech envelope in PPA. I will move on to talking about processing of linguistic representations, and then a couple more methodological things on the reliability of our methods before getting into the ongoing research and future directions. So starting with what is PPA. So PPA, primary progressive aphasia, is a neurodegenerative disorder. It is a network-based neurodegeneration, meaning that the neurodegenerative disease spreads along network lines, for example, within the semantic network or within the phonological network, and is characterized primarily by this progressive decline in speech and language functions with relative sparing of other cognitive processes in the initial stages of the disease. When we're talking about PPA, there are three widely recognized PPA subtypes: the logopenic variant, where these individuals have phonological processing problems; the non-fluent or agrammatic variant, characterized by agrammatism and or apraxia of speech; and the semantic variant, characterized by a loss of core semantic knowledge. And these three PPA subtypes are related to different underlying patterns of neurodegeneration. The semantic variant has atrophy that is in the anterior temporal lobe and spreads posteriorly along the temporal cortex. The logopenic variant in the left temporoparietal region, spreading anteriorly along temporal cortex, and the non-fluent variant in superior frontal and parasylvian cortex in the left inferior frontal gyrus. And these patterns of atrophy are largely predictive of the speech and language deficits that each variant has. So we're looking at speech perception and language comprehension in PPA. So I'm going to present a really basic model of perception and comprehension. So first, you have acoustic input. For example, how can you learn lessons in here? This is mapped onto a spectrotemporal level, which is used in part to transform this continuous representation into discrete representations, maybe something on the order of phonemes or lexical level representations. The lexical representations can be lexical semantic, phonological, or syntactic, before ultimately being mapped onto a semantic representation of the acoustic input. In PPA, receptive language processing has primarily been focused on syllable level, word level, and sentence level processing, with discourse level research in the perception and comprehension domain being relatively lacking. In terms of functional neuroimaging, research has been limited primarily to single syllables and words, and all of this limits to some extent the generalizability to real-world settings. So in the current study, we are looking at using a more ecologically valid approach. So we have participants listen to some sort of continuous stimulus. We'll call this stimulus X. Their brain processes this stimulus somehow, and as scientists, we're recording this activity. In this case, we're using EEG. We take the stimulus and we code it into some sort of feature space. In the example here, I have the speech envelope emphasized in this red contour. And then ultimately, we're working to derive a function that can be used to map that stimulus X onto the recorded EEG. And what we're doing here is trying to estimate what we're calling the cortical tracking of this speech stimulus. And cortical tracking is a term I'm going to use throughout this talk, so I'll give a brief definition now, which is that cortical tracking is how neural oscillations, or brain waves in more colloquial terms, how these neural oscillations are changing as a function of that incoming stimulus. The beauty of the approach we're using is that we can interrogate different levels of processing using the same stimulus. All we do is change the feature model that we're feeding in to the function. And so today I'm going to focus first on tracking of the speech envelope. So the speech envelope contains these quasi-rhythmic amplitude fluctuations, and encoding of the speech envelope involves transforming acoustic input into discrete representations. It has been linked to speech intelligibility and comprehension. So cortical tracking of the speech envelope has been linked to speech intelligibility and comprehension in neurotypical adults. And changes in the speech envelope have also been observed in populations with language disorders. One other bit of methodological detail. So we can look at cortical tracking of the speech envelope in different oscillatory frequency bands, because different bands might be involved at processing different levels of information based on the time scale at which that information unfolds. And so in this study, we're going to be looking at delta and theta oscillations. Delta oscillations occur at about 1 to 4 hertz. That's about 1 to 4 cycles per second, which is about the rate at which we produce words and phrases. And so the delta band has been linked to tracking word and phrase boundaries in addition to prosodic cues. The theta band is a little bit faster, still pretty slow, but about 4 to 8 hertz, so about 4 to 8 cycles per second, which is the rate at which syllables are produced. And here in the bottom corner, I have a modulation spectrum, which shows the peak syllable rate here, and you can see it falls in that 4 to 8 hertz range. So the theta band has been linked to tracking syllable boundaries. So when we're talking about cortical tracking of the speech envelope, bringing it back to that kind of basic model of speech perception, I think probably we're looking at something that's occurring in this acoustic input to spectrotemporal analysis level. Now, granted, I have these bidirectional arrows here because I think it is safe to assume some degree of interactivity among these levels. And so some research has shown, for example, that cortical tracking of the envelope is influenced by this top-down information from these higher levels, but at a basic level, what we're looking at is something around this level here. Okay, temporal response function modeling is the tool that we are using to estimate cortical tracking. And the TRF is basically a proxy to how the brain responds to sound. It involves fitting a linear function to map that stimulus to these temporally precise neurophysiological data, which gives us an idea of how well the stimulus is represented by those neural oscillations. And so bringing back that figure I used before, the temporal response function is the function that we're using to map the stimulus onto that EEG. What you get from this is a predicted EEG. So you use a cross-validation approach, derive predicted EEG, and compare that to the observed EEG. And we look at how correlated that predicted EEG is with the observed EEG. And the more highly correlated they are, the greater the fidelity of the neural representation of that feature. And so here I just have an image of the predicted and observed EEG. Below that is a topographic plot of those prediction accuracies. That's the correlation coefficient between the predicted and observed EEG. So that correlation coefficient across different channels of the EEG. Now, why would we use TRF modeling at all? So TRF modeling, what we've argued and what others have argued is that we're using a more ecologically valid approach by using continuous speech. It allows us to examine neural processing of all of these continuously varying aspects of the speech stimulus. It avoids confounds related to overt responses. So in your more traditional controlled task, you do things like present people with a word or a couple of words, and you ask them things like, are these words the same or different? Like a minimal pairs discrimination task. And part of that is perceiving the words. And then part of it is the decision-making process for determining whether those words were the same or different. And so we have this decision-making confound that's related to overt responses that you can avoid by using this approach. We can also assess multiple levels of representation using a single stimulus, again, just by changing the way that we're modeling the stimulus. And it has shown promise for use in clinical populations. So it's been used in developmental dyslexia and individuals with hearing loss. And so what we're doing in this first part is we are looking at cortical tracking of that speech envelope in the delta and theta range for these naturalistic continuous speech stimuli in individuals with PPA relative to neurotypical age and education matched control group. We have 10 participants in each group, 10 with logopenic, 10 with non-fluent, and 10 with semantic variant, and then 10 neurotypical controls. We have published the data from the logopenic variant in a paper in 2021, so that may be a review for maybe a small minority of people who have read that paper, but otherwise this data is not published yet. So bear with me. We have the left temporal parietal cortex atrophy in the logopenic. We have that superior frontal and inferior frontal and non-fluent, and the anterior temporal atrophy and semantic variant. We have the hearing thresholds mapped over here to show that there aren't differences between the groups. And also I'll say that the kind of mini mental state exam measure of general cognition didn't differ significantly across the PPA groups. So we have relatively matched severity levels, and we have these canonical neurodegenerative patterns in these 3 different groups. We had people listening to 15 one-minute segments of Alice's Adventures in Wonderland. And then after every minute, they were answering 2 multiple choice questions, and they were also given a break of their own chosen duration to avoid any, or to attenuate at least the fatigue that may occur. We collected EEG responses using a 32-channel electrode cap, and we collected these responses continuously while they listened to the story. Okay, so this is a lot. You don't necessarily need to understand this figure to understand the results that I'll be presenting. Understanding generally that cortical tracking is looking at how the brain, how the cortical oscillations are responding to a stimulus. That's kind of good enough. But for those who are maybe interested in knowing some of the more specific details, I'm going to walk through this figure. So we have a speech stimulus here, and we have EEG being recorded while they are listening to that. We take a subset of the data for training the temporal response function. So in this case, we have 15 minutes of story. We take 14 minutes of that as training data. We code that stimulus into some sort of feature space. In this first portion of the talk, I'm talking about the stimulus envelope. We also take that EEG data and we feed that stimulus envelope and the EEG data into a multivariate regularized linear ridge regression. And this is done within the MTRF toolbox developed by Cross and colleagues. The TRF itself, the formula I've got up here, ultimately what we do is we derive that TRF, which you can see here. And we take that left out data, in this case, that minute of data, we take the minute of the stimulus and we use that to derive a predicted EEG, and we look at that relative to the observed EEG. Okay, so we're taking the temporal response function, getting a predicted EEG, correlating it with the observed EEG, and ultimately identifying the prediction accuracy is what we call this. All right, so in this study, we used, like I said, the multi-band envelope, and that is imaged here. We predicted that we would observe impaired performance on those multiple choice questions across all 3 PPA subtypes. Some degree of speech perception deficit has been noted in each PPA subtype. And since the envelope encompasses a lot of different information, we predicted that we would see aberrant cortical tracking of the envelope in all three variants. In the logopenic variant, we predicted that this would be specifically in the theta band, and that's just given the phonological deficit observed in logopenic variant and the fact that the theta band has been linked to that syllable level parsing of the envelope. But we didn't have band-specific hypotheses for the non-fluent or semantic variant of PPA. And again, the way that we are operationally defining cortical tracking is that prediction accuracy between the TRF-predicted EEG and the observed EEG. We analyze these data using a pretty traditional between subjects ANOVA. We did this for prediction accuracy and multiple choice question accuracy, and we used post hoc pairwise comparisons when the omnibus test was significant, in this case, independent samples t-tests. So starting with the narrative comprehension, what we observed was that, and let me orient you to these figures. So on the y-axis is accuracy on those multiple choice questions. On the x-axis are the 4 different participant groups. And what you can see is that the control participants had relatively good accuracy, as did the non-fluent variant PPA participants. In contrast, those with logopenic variant and semantic variant PPA performed relatively poorly on these questions. So that's reflected in this significant omnibus ANOVA and these pairwise comparisons. So for multiple choice question accuracy, for narrative comprehension, impaired in logopenic and semantic, relatively spared in non-fluent. Next, we looked at the prediction accuracy for the TRF on the speech envelope. For all of the cortical tracking results that I'll be presenting, I'll have the delta band on the left, the theta band on the right. The y-axis is going to be the prediction accuracy, that correlation between the predicted and observed EEG. And again, we have participant group on the x-axis. So what we saw within the delta band for that envelope tracking was no significant differences across the participant groups. It was trending that way. It didn't reach traditional statistical significance. The theta band, on the other hand, we do see significantly elevated cortical tracking of the envelope in all 3 PPA subtypes. The omnibus test was significant, and the post hoc pairwise comparisons supported that these 3 PPA subtypes were all tracking at a higher level than controls, but didn't significantly differ from one another. Okay. So what we're seeing here is impaired narrative comprehension in the logopenic and semantic variants of PPA with relatively spared narrative comprehension in the non-fluent variant of PPA. I want to be clear, the use of multiple choice questions is maybe not the most appropriate way of assessing narrative comprehension. This is really kind of a broad brush way of looking at comprehension. But we do think that it is suggestive of a true pattern of impaired comprehension in logopenic and semantic variant PPA. We observed converging evidence for speech perception deficits in PPA. So these deficits have been observed in previous work. And what we're seeing here is that this is manifest as increased tracking in the theta band for every PPA subtype, all 3. Now, again, cortical tracking of the envelope, what we think here at the theta level is that it's involved in parsing the envelope into these abstract syllable level representations. You might think that increased tracking would relate to a better performance or reflect better processing of the stimulus, but actually in the aging literature, it's been argued that this reflects inefficient processing of those cues that are contained in the envelope. And what we think here is going on is that this is pointing to an increased reliance on the speech envelope during continuous speech perception in PPA, perhaps arising from this inefficient processing of acoustic cues. And so perhaps what's going on is that the individuals with PPA are struggling to follow this continuous speech. So they're exerting additional effort. They're focusing as hard as they can on processing this story. And in this case, it seems to be like more at the syllabic level. In the non-fluent variant, perhaps this gets them to normal comprehension or near normal comprehension. But in the semantic and logopenic variant, it's just not enough to get them to that normal comprehension level. Now, to be clear, we're just looking at the envelope here, and actually you can use the cortical tracking approach to look at linguistic representations as well. And so maybe we'll see something more in the linguistic representations to explain what's going on in terms of their perception and comprehension of this continuous narrative. I'm going to shift gears now to talking about cortical tracking of semantic dissimilarity first, because this is where a lot of the research has been focused in the literature. So I'll start by defining what I mean by semantic dissimilarity, which was first reported by Broderick and colleagues in 2018. And so this group, they had individuals listening to a story. They took every word of the story and derived a model of word meaning, in this case using Word2Vec. Word2Vec was used to derive these 400-dimensional feature vectors for each word in the story. And these 400-dimensional feature vectors, it's just a vector of numbers that is argued to reflect some sort of semantic information, and it's obtained basically through web crawling. So taking all sorts of information from the internet and looking at how often words co-occur with each other. That's a very simplified explanation, but I think it's enough for what we're going to be using this for. So you get these 400-dimensional feature vectors, and words that are more semantically similar to each other have more similar feature vectors. So what they're doing here is taking these feature vectors, and in this figure from their paper, what they're doing is taking the current word and looking at how correlated its feature vector is with previous words in the sentence. And then to get dissimilarity, they're subtracting that from 1. So ultimately you end up with a scale of 0 to 2. And the higher the number is, the less similar the words are. So for the, let's say, second word in this sentence or third word in this sentence, you're taking the average of the feature vectors for all preceding words in the sentence. And then they take this semantic dissimilarity value and they feed it into the temporal response function to get an idea of the cortical tracking of that semantic dissimilarity measure. What they found is that this is highly correlated with the N400, suggesting that it is really reflecting some sort of underlying semantic process. And what I have here is an example of semantic dissimilarity for words from this kind of classic N400 paradigm. I take my coffee with cream, and the final word could be sugar or salt or shark or socks or whatever. And what you see is that that semantic dissimilarity value is increasing the further away from the appropriate context, or the less semantically predictable, you could say. All right, now when we're talking about cortical tracking of semantic dissimilarity, here I'm thinking in terms of that model I presented earlier in the talk, that it's probably not just at the semantic level, and that's just because of how these feature vectors are derived. Because we're talking about word co-occurrences, we're talking about more than just semantics. There is lexical and syntactic information as well. So I think we're probably getting at primarily maybe some semantics, but also some lexical level information as well. So what we did is we looked at the fidelity of cortical tracking of semantic dissimilarity in the delta and theta oscillations in individuals with PPA, again, relative to a matched control group. It's the same participants and the same method as what I presented when we were looking at the envelope. So just quickly, this is what that semantic dissimilarity model might look like. In this case, we used GPT-2 to derive our feature vectors rather than word2vec. The main difference here is that GPT-2 provides contextualized vectors. So for an example, the word bank might mean different things when you're talking about money versus a river. In word2vec, the feature vectors for bank would be identical, but in GPT-2, they differ depending on the context. So we looked at that. We controlled for multiband envelope tracking. And what we did is we took a combined model, a TRF prediction accuracy for an envelope plus dissimilarity model, and then subtracted out the prediction accuracy from the envelope. And so here we've got the semantic dissimilarity model with impulses are placed at word onset with a Hamming window. So what do we predict here? Okay, so we expected that we would see aberrant tracking of dissimilarity in the semantic and logopenic variant because of the level at which their deficits occur at the phonological and semantic level. We thought this would probably be in the delta band because this is a word level model, and we thought this would reflect impaired processing of lexical and semantic representations. We didn't predict a difference here for the non-fluent variant, although arguably you might predict that. Okay, statistical analysis, same analysis as before. I'm not going to show multiple choice question accuracy because we used the same data as before, and instead I'll jump right into the cortical tracking results. Okay, so what do we see here? We're not observing any significant differences between our PPA groups and controls, nor are we observing any differences between each PPA group. So that's true at the delta band level and at the theta band level. Okay. So ultimately, we're not seeing significant differences in tracking of semantic dissimilarity. We showed this previously using word2vec. We were hopeful that GPT-2 might give us a different outcome, and in fact, we see the same findings in both word2vec derived feature vector models and GPT-2 derived feature vector models. Now, semantic dissimilarity has been fairly widely used to evaluate semantic processing, but it's primarily been in younger adults. For us, we were ultimately unsuccessful in differentiating older adults from PPA using this specific approach, but it's also been shown more recently that older adults may not respond to semantic dissimilarity the same way that younger adults do. There's also really recent work suggesting that semantic dissimilarity may not add predictive power over the envelope. So perhaps some of these previous studies that have shown significant tracking of semantic dissimilarity, not all of them controlled for the speech envelope. And the speech envelope is something that we controlled for in the current study. The same study that found that semantic dissimilarity didn't add predictive power over the envelope did show other linguistic variables adding predictive power. And we didn't look at all of the variables they showed, but I'm going to now show you, we looked at a couple of those variables that they showed adding value over the envelope. And in a different study, let me take a step back. So the linguistic variables that we are going to look at from this Gillis et al. Study are cohort entropy, which is believed to lie at the sublexical or lexical phonological level, and word frequency, where we're looking at lexical semantic processing. These map on very well to the deficits observed in logopenic and semantic variant, respectively. But we also wanted to look at a model that would map onto the deficit observed in the non-fluent variant. And so what we did here is there's another study that came out more recently that found that syntactic surprisal added power over the envelope. So we also looked at syntactic surprisal. So for cohort entropy, what this value or what this model is believed to reflect is how much competition exists between all of the words that are in the current words cohort up to the current phoneme. And so the cohort reflects all of the words that share that same acoustic input up to the current phoneme. And argued to reflect some sort of sublexical phoneme or phonological level processing. When we look at cortical tracking of cohort entropy, again, we're not seeing any differences between groups in the delta band or in the theta band, which you might expect cohort entropy to be linked more closely to the theta band, given the theta band's role in that smaller level of processing at the syllable level, but not seeing any differences here. And in fact, if you look at the y-axis, this is the difference between the combined envelope plus cohort entropy minus envelope. And so if anything, we're seeing that adding cohort entropy maybe was adding noise to that model because it leads into these negative values here. For word frequency, this is just how frequently a word occurs in a language. We see a similar pattern here where there aren't differences between groups. At the delta level, we're at least seeing some added value of adding this to the envelope, but not seeing differences across the 4 participant groups. And at the theta band, similar outcome, no real differences across the groups. All right, lastly, we looked at syntactic surprisal. This is how surprising the part of speech of the current word is given the distribution of parts of speech for preceding words. And again, we're not really seeing any significant differences between groups at the delta level. In the theta band, we are seeing maybe a trend here in the logopenic, but we're still in the negative range here for the added value of syntactic surprisal, so I'm hesitant to make too much of this. Now, lack of significant differences in any of these linguistic models for cortical tracking. There are a couple of things we're doing now that are a more nuanced approach to interpreting these data. So one, prediction accuracy is averaged across the entire length of the stimulus, but the temporal response function is giving us this time course information. And so we are imagining a situation where the TRF time course is going to provide a more nuanced representation of the processing of the stimulus, because the TRF can inform the time course of the neural dynamics that underlie the prediction accuracies that I've presented so far. The other thing here is that I'm reporting prediction accuracy averaged across all EEG channels. But in fact, other more recent work has shown that you might actually, or what I'm doing by averaging across all channels is I might be wiping out the actual effect that we would observe in some key channels that are more involved in processing that representation. And so here I have a screenshot of a figure from a recent study. Here is plotting the change in prediction accuracy for some linguistic representation. And you can see that some of these channels are in that negative range. And so perhaps when we're averaging across all of the channels, we are wiping out the effect that we might see. And so now we're looking at the channel level prediction accuracies as well. Now, in terms of the temporal response function, in terms of the time course, I haven't analyzed these data, and clearly this figure is a really rough figure. It doesn't have error bars or anything like this, but I wanted to just give an idea of the time course. So this is for semantic dissimilarity in the delta and theta band. And what you can see, or at least what I think you can see, is that there are differences in the time course of processing, with some peaks being delayed. And so perhaps now in our next step, where we're looking at these temporal response functions themselves, this is where we may begin to see these differences. So we're going to start looking more closely at the channel level data and at the time courses themselves. All right. Now, despite the lack of significant differences in prediction accuracy, we have used these TRFs for a different kind of approach to examining differences across groups. And so we think that the TRF may be useful for informing differential diagnosis by PPA subtype. In this case, we're doing machine learning classification using the temporal response function itself. This is work that has been spearheaded by a student I've worked with at UT, Lokesh, who did all of this for their senior honors thesis. And so I have in this first column, the different classification tasks, so healthy controls versus PPA, and different comparisons between PPA groups themselves. We were showing the best model here. So we ran models for all of these different linguistic representations, as well as the envelope. We used a variety of classification algorithms. And what I have here is the outcomes for the best model balancing precision. So that's the proportion of true positive cases that are predicted as positive over all predicted positive cases, as well as recall, which is the proportion of true positive cases predicted as positive over all of the true positive cases. So F1 balances these two. And you can see here a range, like the worst model that we have is this three-way differential diagnosis by PPA subtype. But I really want to draw your attention to this non-fluent versus logopenic one. So traditionally in the clinical domain, these two PPA subtypes are the hardest to differentiate from one another. And in fact, in the initial criteria laid out by Neary and colleagues in 1998, non-fluent and logopenic were encompassed by a single category, progressive non-fluent aphasia. And so we think it's really promising that we're getting such good F1 scores for what is clinically perhaps the most difficult to distinguish. And so this work I am just presenting very briefly. I'll be presenting it at the Clinical Aphasiology Conference in May in more detail, but more or less what I want to show here is that all hope is not lost for the TRF approach. We still have the time courses to look at. We still have the channel level prediction accuracy to look at. And we're showing here that the TRF itself is actually quite useful for differential diagnosis by PPA subtype using a machine learning classification approach. Okay, now maybe this is something I should start with to show that these measures we're extracting are reliable, but it is something we are just now looking at since I started at U of H about a year and a half ago. So here we are basically saying we can collect these measures and use them all day long, but if they're not reliable, what good are they actually going to do? If they're not reliable, if they're not valid, what value do they actually add? So when looking at test, retest reliability, we've done this in neurotypical young adults and have preliminary findings in stroke-induced aphasia. And I'm just going to show a little bit here because we're getting close to time. But here I have had undergrads coming in for 2 different sessions separated by at least a week. And they're listening to Who Was Albert Einstein, which is a story that was used by Stephen Wilson, validated for use in stroke-induced aphasia that we've been using both in PPA and in neurotypical young adults and individuals with stroke-induced aphasia. We have their session 1 prediction accuracy on the x-axis and session 2 on the y-axis. In this case, I'm showing you the broadband envelope derivative. This just emphasizes acoustic onsets and offsets. I'm reporting in the theta band here, but I hope that you can appreciate how strong this correlation is across sessions. So we have an intraclass correlation coefficient of about 0.85, which is pretty strong and pretty, it gives me good confidence that at least in a neurotypical younger adult population that these measures are reliable over time. We also looked at this in stroke-induced aphasia and have ongoing data collection. Looking at this, I just have 2 participants now, participant 1 on the left, participant 2 on the right, session 1 and session 2 for each with prediction accuracy on the Y. And what I hope you can appreciate is that these are also looking very reliable over time. And so this, to me, is quite promising, potentially indicating that these models not only are reliable, but also could potentially be used to look at neural changes in response to treatment. So we have lots of ideas for how we can use these models in the future. One thing I want to note, there's a recent preprint that has used cortical tracking in stroke-induced aphasia. I do want to say that their patterns in stroke-induced aphasia appear to differ from what we've observed in PPA. So in PPA, we're seeing increased tracking of the envelope. What this study showed was reduced tracking of the envelope. And actually what we're seeing here across these two participants are kind of two different patterns. And so I'm really interested in looking at how these patterns change as a function of the type or patterns of deficits in stroke-induced aphasia. So for that, I'm showing that these measures are pretty reliable. And what we're doing now is we're examining the time course and channel level prediction accuracies. We're working on additional linguistic models and refining the existing models that we're using. We're working to identify other ways to control for acoustics. So there are a variety of approaches that you can use. Here we've used a pretty basic subtraction approach, but you could do things like predict the TRF with the envelope, save out the residuals and feed those into a subsequent TRF with linguistic representations. And there are other ways we might be able to control for acoustics as well. And lastly, I'm really interested not just in examining how reliable the TRF is over time, but looking at how it relates to these more traditional constrained tasks that are used more in clinical practice. And so seeing whether there is a good mapping between these two traditional tasks and these TRF-derived measures. So that brings us to the end. I have to thank my funding sources. So a lot of this data was collected during my time as a postdoc with my F32, my two sponsors, Maya Henry and Bharat Chandrasekaran, as well as funding from Dr. Gorno Timpini, and then some of that reliability stuff that's been more supported by my startup funds since I started at the University of Houston. I want to thank you for your attention, of course, and all of the mentors and collaborators I have. I've emphasized those who were most involved in the data I presented by bolding and underlining them. I want to draw specific attention to Nike Gananateja, who has been working very closely with me, or rather I've been working very closely with him on all of these TRF approaches. And then my new lab since I started at U of H, who are just the best and brightest, and I'm really fortunate to have such a good group to work with. So lastly, but certainly not least, thanking the participants with PPA and their care partners, the participants with stroke-induced aphasia and their partners as well, for participating in this study and helping us to learn more about perception and comprehension in aphasia. And so that brings me then to the end of my talk. So thank you.
Thank you, Heather. That was great. I hope you could hear the applause in our room here. So thanks a lot. That was a really nice talk. Let's open it up first. I'm going to open it up for questions in our room. See if anybody... I don't see any hands. So if that's the case, then I can move to the questions that are online. First was a question of clarification, I think from Alex Craig. Are the predicted observed EEG channel averages? And if so, or which channel? And and he says, oh, I suppose the results in correlation coefficients are per channel.
That's right. And then we averaged across all of the channels for the box plot data I presented, but we're looking at that channel level data now.
Yeah. Okay. We have a question from Greg Hickok. Is comprehension and envelope tracking correlated? I think he means are comprehension and envelope tracking correlated?
Yeah, so a lot of work has suggested that they are, that increased tracking of the envelope is linked to comprehension. There actually may be this kind of inverted U-shaped relationship where up to a certain point, increased tracking is good for comprehension. And then after it reaches a certain point, it falls off and increased tracking relates to poorer comprehension. So the relationship may not be linear, but it does seem to exist. Now, I just read a journal preprint yesterday from Peter Hogwart's group that's suggesting that a lot of studies have kind of confounded intelligibility with comprehension. So whether you can understand the word, like if you're listening to noisy stimuli, can you tell me what word you heard? Which is not the same thing as understanding that word, right? Like, so the perceptual ability to perceive the phonological form of the word versus understanding the meaning of the word. A lot of the previous work has kind of confounded these two things, but kind of a short answer is that they do seem to be correlated, although maybe I think there are still some questions we need to answer there.
Okay, thank you. Question from Matt Davis. Was cortical, so this I think specifically relates to the studies where you, the later studies that you showed where you didn't have differences, right, between the groups. So Matt asks, was cortical tracking of semantic similarity greater than chance in the control participants?
In a small subset, but not in all of them. And in fact, I think that is kind of the issue we're showing with the negative added value of these linguistic representations, that perhaps we're really just adding noise, at least the way that we're looking at it right now. So short answer, as a whole, no, not greater than chance.
Okay, thank you. I had a question. I may have missed something in your explanation, but I'm wondering about the delta band. And it's you did mention, well, yeah, there are suggestions that there are higher language level functions come into play, like prediction maybe, but you didn't really go into detail about that. It would seem to me that delta tracking of word boundaries and phrase boundaries is by definition requires this higher processing, right? That's not just prosody. So I basically have two questions about that. One is, isn't it the case that delta band processing should by definition be dependent on successful theta band processing? So if you're not tracking syllables, how would you get to the word level? But also, I was wondering how you establish the delta band target or source function. Is it just based on acoustics, or are you applying a manual segmentation or something there in words or phrases? I think I missed that.
Let me clarify, actually. So we are filtering the EEG data into the delta and theta bands. The stimulus model going in is the same regardless. And so we're using just a standard EEG filtering pre-processing approach to that.
So you're not actually looking at using the speech signal itself. Right.
Oh, now, okay. Some people have used the rate of word production and syllable production in their stimulus to then define their delta and theta ranges in their EEG data. We used a more basic approach of just filtering the EEG into 1 to 4 hertz and 4 to 8 hertz. But I think your other question is a really interesting one. Shouldn't processing of words rely on processing of those lower level representations, especially if we're thinking of a strictly hierarchical model or even a hierarchical model with interaction like I presented? And, I think this is really interesting. So in a few studies that have come out more recently, there's some indication that perhaps when we're using more constrained stimuli, things are really more incremental than when we're using more continuous stimuli. And so some work, like I think some of the stuff from Laura Williams has suggested that maybe these things are being processed in parallel. So these phoneme or sublexical level representations are being processed in parallel with the higher level word level representation when you have continuous speech. Versus if you have more constrained controlled stimuli, you might actually be using more of this incremental hierarchical approach. For what we're doing, I think perhaps it's a different issue since we're filtering the EEG data, but it's something that, or rather, I think I want to think about it a bit more and try to think about how the filtering of the EEG data and looking at it in that way. Actually, you know what? Okay. So I think the time course is really where this comes into play. So when we look at the TRF time course, we may be able to see a little bit more about how these things are relating to each other across the different frequency bands. I still want to think about it a little more.
Oh, thank you very much. A question from Corinne Burstis. You mentioned removing the confound of the decision-making process in your listening tasks, but are there remaining confounds to potentially consider, like whether the participant was spacing out or sleeping?
Oh, yeah, for sure. Those confounds exist. We try to minimize them by giving breaks every minute, by allowing people, like, if they needed to get up and walk around, we could disconnect the channels, tuck them in a little hood. We usually tell people to wear a hoodie, tuck the channel cables in a hood and let them walk around to wake up a little bit. We try to keep the task relatively short, so only about 15 minutes of story, with larger breaks in between stories. But absolutely, this is a concern. This is a concern that I think one of the reasons why I really wanted to look at reliability was because I suspected that this could play a role. Like, if you're especially sleepy one day, but not the next day, would this affect tracking of these different linguistic or acoustic representations? And I think the answer is probably yes. But what we're showing so far is that if those differences do exist, they're having minimal effect. Now, I know that doesn't really answer your question. So there are certainly additional confounds to consider. We're hoping by design of the study that we've minimized them to some extent.
Great, thank you. Question from Benjamin Zinser. Comparing the TRFs between PPA variant groups, are those temporal response functions all estimated with the same tuning parameter, lambda, usually optimized post hoc? And possibly the classification task could work on just which lambda value yields highest correlations before looking at the estimated TRF itself.
Yeah, so for every person, we selected the TRF for the lambda that had the best prediction accuracy. So there's the same range of lambdas that we use across participants, but we select the one that was best for that person. So at an individual participant level, I have not considered feeding that into the classification task. Is a really interesting idea. I'd love to talk more offline about that.
Great. We have another question from Matt Davis and a comment. Great talk, Heather. Thank you. Can you say more about the challenges of collecting EEG data in these groups? Studies with younger or healthy volunteers typically collect more than 15 minutes data from each participant. Would this be feasible for PPA patients?
Yeah, so we actually have two different narratives. I only reported on one of them. We have 30 minutes total across the two narratives. In the 2021 paper, we showed that at least within the logopenic variant and older adults, that tracking across two narratives was highly correlated. So it was really reliable across narratives that differed in a number of acoustic and linguistic features. But I think always more data is better. We do run up against the risk of fatigue. So like Corinne's question a little while ago, we're trying to minimize that possibility by keeping it relatively short. Now, with both PPA and stroke-induced aphasia, I've found that every participant we've recruited has been able to complete the task, and I've only had one person fall asleep during the testing, and her data I don't have included in any of the analyses I reported today. This was an individual with stroke-induced aphasia. So there are some additional considerations. We've tried to build that into the task, but also, yeah, we're not collecting an hour of story in these people the way some other people have. Yeah.
Great, thanks. And what I think is our last question from Maya Henry. You mentioned that there may be differences in semantic dissimilarity effects in younger versus older individuals. Now that you have data from younger adults, undergrads, do you plan to look at this and other linguistic models in younger versus older groups?
Yeah, that's such a great question. We are, looking at aging effects in a number of the studies we're conducting right now that I didn't report. And so I think it is a natural next step for us to also do that here. Now, I'm trying to collect older adult control data on the specific stimuli we're using in younger adults. There are some, we collected some similar data at UT and at U of H, but it's not identical. So once I get a bigger control group of older adults here at U of H, that is absolutely something that I want to look at. Thank you.
Great. And I think that brings us to 2 minutes before 1 o'clock. So perfect timing. Thank you so much, Heather, for your talk. Thank you, audience, for your participation. And we hope to see all of you again in 2 weeks' time for Jackie Hinckley's talk. Bye-bye.
All right.